Today we discussed about meiotic cell division, that is also known as meiosis. It is also as reduction division. It is also known as reduction division. The chromosome number of mother cell, that is always 2n. The chromosome number of mother cell always become diploid, that is 2n, is reduced to half in daughter cells, that is in gametes. The meiotic division is also known as reduction division because chromosome number of mother cell, that is always diploid 2n, is reduced to half in daughter cells. That means the gamete is n haploid. Meiosis occur in reproductive cells of reproductive cells of plants and animals. Meiosis, reduction division occur in reproductive cells of plants and animals, that is male and female sex organs. In plants, it occur in pollen mother cell of anther and megaspore mother cell of ovary. The meiosis or reduction division occur in plants in pollen mother cell of anther and megaspore mother cell of ovary. The pollen mother cell and megaspore mother cell. Here the meiosis occurs and the gametes are produced. Pollen grains are the male gametes, that is the end product of meiosis in pollen mother cell, and egg cell is the female gamete, that the end product of meiosis that took place in megaspore mother cell of ovary. In case of animals, the reduction division or meiosis occur in testis in case of male and ovary in case of female. The gametes are sperm gametes, male gametes, sperm, and the female gamete is egg cell. Then next topic, what is the importance of meiosis? Importance of meiosis. What are the importance of reduction division or meiosis? The number of chromosomes remain constant in a species generation after generation. The number of chromosomes remain constant in a species generation after generation. That is the chromosome number of onion, that is 16, but the chromosome number of gamete, that is 18 number. In case of potato, the chromosome number 12 in somatic cell or reproductive organ, that is 48, but in case of gamete, that is 24. Horse 64 to reduce in gametes 32, and in case of man the gametes occurred only 23, but in somatic cells it is 46. 2n is 46 and n is 23. Second point, the importance of meiosis. The meiosis occurs during gamete formation, that is known as gametogenesis. The gamete formation is known as gametogenesis, male gametogenesis and female gametogenesis, and reduce the number of chromosomes from diploid to n to haploid, that is n, in the gametes. These diploid gametes fuse to form diploid. These haploid gametes fuse to form diploid zygote during fertilization. It occurs, that is, the meiosis occurs during gamete formation The gamete formation is known as gametogenesis. And reduce the number of chromosomes from diploid that is 2n in gametes that is to haploid in gametes. These haploid gametes fuse to form diploid zygote during fertilization. Haploid gametes, that means haploid male and haploid female gametes fuse to form diploid, that is 2n in zygote during fertilization. Third point, meiosis establishes new combination of characters, that means variation. Meiosis establishes new combination of characters due to mixing of paternal and maternal chromosome and crossing over during prophase 1. That is very important. Meiosis establishes new combinations of characters due to mixing of paternal and maternal chromosomes, that is the chromosome from father and mother, mixing of paternal and maternal chromosomes, that is one source of this combination or variation. The second source for variation, that is crossing over. Crossing over during prophase 1. During prophase 1. There are two chances for variation, that is the mixing of paternal and maternal chromosomes, and also the second scenario, that is the crossing over, that is one of the phase in prophase 1, that is packaging stage, that also cause the variation. Variation is the different character that is seen by the individuals from their parents, that is variation. Variation is the basic unit for evolution. The meiosis establishes new combinations of characters due to mixing of paternal and maternal chromosomes, and second, crossing over during prophase 1. As a result, the progeny, that is the individual or child, get the traits of both the character of mother and father in new generation or new combination. As a result of variation or this type of mixing, the progeny means young ones or children. The progeny inherits the traits. Traits means characters. The traits of both the mother and father. In new gene combination. New gene combination. Now we draw a flow chart of meiosis, the steps of meiosis that we include and post in a flow chart. First, meiosis. The meiosis with two main steps, that is meiosis 1 and meiosis 2. Meiosis 1 and meiosis 2. The meiosis 1 with 4 steps. The meiosis 1 with 4 steps. That is prophase 1, metaphase 1, anaphase 1, telophase 1. The meiosis 2 also with 4 steps. The meiosis 2 also with 4 steps, that is prophase 2, metaphase 2, anaphase 2, telophase 2. Meiosis with the main 2 steps, meiosis 1 and meiosis 2. Meiosis 1 with 4 steps, prophase 1, metaphase 1, anaphase 1 and telophase 1. The meiosis 2 also with 4 steps, that is prophase 2, metaphase 2, anaphase 2 and telophase 2. This prophase 1 is one of the longest step in meiosis 1. Prophase 1 with 5 substages. The prophase 1, that is the longest phase in prophase 1 or altogether meiosis, that is liptotin, zygotin, pachytin, diplotin and diakinesis. The meiosis with meiosis 1 and meiosis 2. Meiosis 1 and meiosis 2 with 4 steps, prophase, metaphase, anaphase, telophase 1 and prophase 2, metaphase 2, anaphase 2 and telophase 2. This prophase of meiosis 1, it is the longest phase in this reduction division, that with leptotin, zygotin, pachytin, diplotin and diakinesis. Next, we discuss about meiosis 1. First we discuss about all steps of meiosis 1. Prophase 1, metaphase 1, telophase 1, anaphase 1 and also the sub stages of prophase 1, liptotin, zygotin, pachytin, diplotin and diakinesis. We include this meiosis 1 in this part, and in next session we discuss about meiosis 2, that is the different steps of prophase 2, metaphase 2, anaphase 2 and telophase 2. Now we discuss about meiosis 1. Meiosis 1. This meiosis 1, that is the first meiotic division, that is also known as first meiotic division. It is a longer phase. It is further subdivided into the following 5 substages. It is a longer phase, that is meiosis 1 is a longer phase. It is further subdivided into the following 5 substages. First, liptotin. What happened in liptotin? The chromosome become distinct and appear as long and thin threads. Now we draw the nucleus, not the cell. The nucleus of a cell. Here This is the nucleolus and we draw 6 chromosomes. We take 6 chromosomes as an example. What happened in liptotin? The chromosome become distinct and appear as long and thin threads. In early stage, the chromosomes are not formed but chromatin reticulum are seen in, that is thread-like chromatin reticulum are seen in a younger stage of the cell. But at the time of liptotin, that is the starting of meiosis, the chromatin reticulum condensed to form chromosomes. The chromatin reticulum condensed to form chromosomes. And appear as long and thin threads. Appear as long, thin threads. Then each chromosome consists of 2 chromatids. Each chromosome consists of 2 chromatids. Chromatids are the arms of or half pieces of a typical chromosome. Each chromosome consists of 2 chromatids held together by a centromere. This is the centromere. This is the centromere. These are the chromatids. This is the centromere. Each chromosome consists of 2 chromatids held together by a centromere. Next, the nuclear membrane and nucleolus are distinct. That is clear. This is the nuclear membrane and this is nucleolus. The nuclear membrane and nucleolus are distinct or clear. Now complete the main 3 steps of liptotin. Next, zygotin. Zygotin. What happened in zygotin? The chromosomes continue coiling and become shorter and thicker. Now we draw the picture. The chromosome continue to coiling and become shorter and thicker. that is chromosome condensation continues. Shorter and thicker. The homologous or similar type or size of chromosomes start pairing from one end. This pairing is known as synapsis. This chromosome coming from father and this from mother. This from father and mother. The homologous chromosome, that means similar type of chromosome, start pairing from one end to another part end. This pairing of homologous chromosomes are known as synapsis. Pairing of homologous chromosomes are known as synapsis. This pairing is known as synapsis. The third step of zygoting. Each pair of homologous chromosome is called bivalent. Each pair of homologous chromosome is now called bivalent. Each pair of homologous chromosomes now called a bivalent. Then nuclear membrane and nucleolus are distinct. That is clear. Nuclear membrane and nucleolus are distinct. That means clear. Now complete the stages of zygoting. Chromosome continue to coiling and become shorter and thicker. The homologous or similar chromosomes pairing, that is known as synapsis. Each pair of homologous chromosome is now called bivalent. The nuclear membrane and nucleolus become distinct or clear. Next step, pachytene. The chromosome become shorter and thicker. The coiling continue due to further coiling. Each paired unit called bivalent shows 4 chromatids. Hence, the bivalents are also known as tetravalent. Now, the replication formed. So, each bivalent become tetravalent because of replication. the bivalents become tetravalent by pairing of bivalent shows 4 chromatids. So each chromosome with 4 chromatids. Here only 2 chromatids, but here 4 chromatids. Now write the points. The chromosome condensation continues. The chromosomes become shorter and thicker due to further coiling. Next, each paired unit called bivalent show 4 chromatids by replication, and hence bivalence is known as tetravalent. The bivalents become tetravalent. They produce one copy of this bivalent and to form a tetravalent. Now this chromosome with 4 chromatids. Tetravalent. Here a replication or produce a same copy of bivalent and form a tetravalent. That means the replication occurs. Then crossing over occurs. Crossing over occurs at the end of packaging, that is break and exchange of parts, that means genes of chromosome occurs between non-sister chromatids. These are the non-sister chromatids. These are the non-sister chromatids. This is this chromosome from father and this from mother. So it is known as non-sister chromosomes. Non-sister chromosomes with non-sister chromatids. The crossing over always took place between the chromatids of non-sister chromosomes. Now we draw the another picture that shows the crossing over. Here we can see one crossing over, and that means the exchange of chromatin material, that is genes, between the paternal and maternal chromatids or genes. The genes are the part of DNA. DNAs are the part of chromosomes. So gene parts are exchanged between non-sister chromatids. This crossing over may be 1 or 2 or 3 points. Now we only draw 1 point. Here one crossing over there, and also here one crossing over occurs. This occurs at the end phase or end part of packaging. The crossing over, that is the exchange of chromatin materials or gene parts between non-sister chromatids. The non-sister chromatids are the chromatids of the homologous chromosomes that arrive from the father and mother. Crossing over occurs at the end of packaging stage, that is crossing over, that is break and exchange of parts, break and exchange of parts. Parts means genes. Occurs between non-sister chromatids of a homologous pair, The point of interchange and rejoining. The point of interchange and rejoining appear as X shape and is also known as chiasma. The plural chiasmata, or the point of crossing over. The point of interchange and rejoining the point of interchange and rejoining appear X shape and is known as chiasma. This X shape is known as chiasma. The plural of chiasma, that is chiasmata, or the point of crossing over. Now complete the different steps or parts of packaging. That is very important. Here the chromosome materials are exchanged. That is the basic of variation. That is the difference between the parents and young ones. Next, diplotin. Diplotin. What happened in diplotin? The chromosome continues coiling further and become shorter. The coiling continues. The centromeres of homologous chromosomes start repelling each other. Now draw the picture. The chromosome continue coiling. And shorter and thicker. Second, the centromeres of homologous chromosomes start repelling each other. The centromeres of homologous chromosomes repel each other. Repelling. Then the two non-sister chromatids of a homologous pair of chromosomes retain attached at one end or 2 points, that is chiasmata. The attachment only seen in chiasmata. Third point, the two non-sister chromatids, non-sister chromatids of a homologous pair of chromosomes remain attached at one or 2 points. We only shows one chiasmata, not two. One or 2 points. That is the chiasmata. Then nucleolus and nuclear membrane become indistinct. This dissolved in cytoplasm. This start the nuclear membrane and nucleolus try to dissolve in cytoplasm. The nuclear membrane and nucleolus indistinct, not clear. Nucleolus and nuclear membrane start to dissolve or disappear. That means indistinct. Indistinct. When it is at the chiasmata, that exchange of non-sister chromatids, that is genes, between homologous chromosomes has taken place. The process of gene exchange is known as genetic recombination. The process of such genetic material exchange is known as genetic recombination. This process of gene exchange is known as genetic recombination. Next, diakinesis. What happened in diakinesis? The bivalents become the shortest and thickest due to maximum coiling. The bivalents or chromosomes become shortest and thickest due to maximum coiling. The centromere, that the joining part of chromatids, central part, the centromeres and non-homologous parts of homologous chromosomes of a bivalent move apart due to repulsion from each other. Here is the point of recombination. These are the points that the gene exchange parts. We typically represent this, the structure or the stage. The bivalents become thicker and shorter due to maximum coiling. Maximum coiling of chromosomes or bivalents or chromosomes or chromatids occurs then the centromeres and the centromeres and non-homologous part of non-homologous parts of homologous chromosomes. Of a bivalent move apart due to repulsion from each other. Then nuclear membrane and nucleus completely disappear. Nucleus and nuclear membrane completely disappear. These are completely disappear, and these recombinant DNAs or chromosomes freely seen in the cytoplasm. The nuclear membrane completely dissolved and chromosomes are dispersed in cytoplasm freely. The spindle formation is started. Here spindle formation is started. These are the spindle fibers that is arise from a cell organelle, centrosome. The spindle fibers are arise from the cell organelle centrosomes, not centromere. This is centromere and centrosome are the cell organelle like mitochondria, ribosome, etc. The spindle fibers are forming to start. Spindle formation started. Now complete the 5 steps of prophase 1. Next, metaphase 1. Metaphase 1. There are 5 detailed steps of prophase 1. Then metaphase 1. The next step of meiosis 1 is metaphase 1. Metaphase 1. The bivalents arranged themselves at the equator plate. That means the central part of the cell or cytoplasm. This is the cell, not the nucleus. The chromosomes dispersed in the cytoplasm, nuclear membrane and nucleus disappears. This is the plasma membrane, not the nuclear membrane. What happened in metaphase 1? The bivalence. The bivalence. Arranged themselves at the equatorial plate, that is a central plate. Plate means space. The bivalence arranged on the central part of the cell. Then second point, spindle fibers attached at the centromere of chromosomes. The spindle fibers attached to the centromere of the chromosome. One centromere of a bivalent is joined to one pole and the second centromere is joined to opposite pole. By separate spindle fibers. One centromere of a bivalent is joined to one pole and second centromere is joined to the opposite pole by the separate spindle fibers. By the separate spindle fibers. Now complete the metaphase 1 stage. Then next anaphase 1. Anaphase 1. The spindle fiber shorten. The centromere of homologous chromosomes are pulled along by the spindle fibers towards the opposite poles. That is no division of centromere. Now we draw the picture. These are the spindle fibers. The spindle fibers shorten. The polar spindle fibers shorten. And some spindle fibers are erased in between these chromosome or the equator part also. The spindle fibers become shortened. The centromere of homologous chromosomes. The centromere of homologous chromosomes these are the homologous chromosomes. The centromeres of homologous chromosomes are pulled along by the spindle fibers. Towards the opposite poles. No division of centromere occurs. That is important. No division or partition of centromere. Third point. Thus, half of the number of chromosomes, each with 2 chromatids of the parent cell, go to one pole and the remaining half to opposite pole. Thus, half of the number of chromosomes each with 2 chromatids of the parent cell go to one pole and the remaining half to opposite pole. The partition half become move to one pole and another half to another pole. Then the fourth point, each set of chromosomes that move to poles consists of a mixture of paternal and maternal chromosome parts. This we did pictorially represent the part, the mixing part. Each set of chromosomes that moves to poles, different poles, consist of a mixture of paternal, that means father, paternal and maternal chromosome parts. That means new gene combination, the chromosome with new gene combination. Then the last part of prophase 1, that is telophase 1. Telophase 1. What happened in telophase 1? The separated chromosomes uncoil in the newly formed daughter nuclei. There are two nuclei are formed in the parent cell. This is the cytoplasmic part. Now, two nuclei are formed in a single cell. The separated chromosomes uncoil in the newly formed daughter nuclei. There are two nucleus, that is known as nuclei, in the same parental cell. Then the daughter cells now have half of the amount of DNA as compared to that of anaphase. There are 6 chromosomes now become half in number, that is 3 in each nuclei. The daughter cells now have half the amount of or number of DNA or chromosomes. DNA are the part of chromosome. DNA are made up of genes. DNA or chromosomes as compared to anaphase 1 or the parental cell, that means the first parental cell. The nucleolus reappear and nuclear membrane forms. The nucleolus reappear and nuclear membrane forms. Reappear. Then the daughter nuclei enter into the second meiotic division. The daughter nuclei, these daughter nuclei enter into the second meiotic division, that is meiosis 2. In next session we discuss about meiosis 2. Now we complete all steps of meiosis 1 very detailed with pictures and points very clearly. Then next session we discuss about the second part of meiosis, that is meiosis 2. If you like this session or class, please like, share and subscribe our channel.